Forensic Science Lecture 12-1, Forensic Examination of Hair. Examination of hair evidence. We know that trace evidence often corroborates other evidence developed during the investigation. Hair is a common type of evidence recovered at a variety of crimes, but as of yet, it's not possible to individualize any single head or body hair to an individual, but it's still valuable as physical evidence. They have had partial success of individualizing with the advancement of DNA analysis. When properly collected and submitted with adequate number of standard reference samples, it can provide strong corroborating evidence. So let's look at the morphology of hair, or the structural characteristics of hair. Hair is considered to be an appendage of skin that grows out of an organ that we call a hair follicle. So it's like an arm, an ear, a leg. It's an appendage. It extends from its root or bulb embedded in the follicle and continues into a shaft and terminates at a tip end. There are two features that make hair a good subject for establishing individual identity. One, its resistance to chemical decomposition and its ability to retain structural features over a long period of time. The shaft is what is mostly examined by forensic scientists, and is composed of three layers: the cuticle, the cortex, and the medulla. We look at the morphology of the cuticle first. These are the scale structures that cover the exterior of the hair. Much of the resistance of hair to the chemical decomposition is contributed to the cuticle. It's formed by overlapping scales that always point to the tip end. It's formed by specialized cells that have hardened and flattened as they move from the follicle. Scale patterns is useful in species identification as well. There tends to be three patterns: coronal or crown-like. These resembled stacked paper cups. Spinous or petal-like, which are triangular in shape and protrude from the shaft. And then imbricate or flattened, and these consist of overlapping scales with narrow margins. The scale patterns aren't useful for characterizing individuals, but like I said, they're very useful for identifying species. Moving on to the cortex, this is the main body of the hair shaft. It's made of spindle-shaped cortical cells that are aligned in a regular array, parallel to the length of the shaft. The major forensic importance is that it's embedded with pigment granules that give hair our color. And so color, shape, distribution of these granules all provide criminalists with important points of comparison when comparing a known hair to the crime scene hair. The morphology of the medulla. This is the cellular column running through the center of the hair. It can look like a central canal running through the hair. It's a prominent feature in most animals. It occupies generally 1/2 or greater of the diameter of shaft of most animals. In humans, however, it occupies less than 1/3 of the diameter. The medullary index measures the diameter of the medulla relative to the diameter of the hair shaft. And so this is how forensic scientists will, or what they will use in order to compare the known reference sample to the sample from the crime scene. We can also look at the presence or the absence of the medulla, because that varies from individual to individual. It even varies among hairs of individuals. So humans and other animals have nearly cylindrical shaped medulla. Cats have been described as resembling a string of pearls, um, but they can become classified into the degree of the medulla. So continuous, interrupted, fragmented, or absent altogether. Um, uh, this also can be looked at the different shapes of them depending on the species. Like I said, with the cats and this and the looking like pearls, strings of pearls. But there's a database that exists of 35 most common animal hairs encountered in forensic cases. And this allows examiners to rapidly search for animal hairs based on scale patterns and medulla types. Moving on to the root morphology, the root and the surrounding cells provide the tools necessary to produce hair and continue its growth. And there are 3 phases of hair growth which determine the shape and the size of the root: anagen phase, catagen phase, and telogen phase. If we're looking at the anagen phase, this is the initial growth phase in which the follicle is actively producing hair. It can last up to 6 years. The root is attached to follicle, giving a root bulb a flame-shaped appearance. And when pulled from the head, some translucent tissue surrounding the hair follicle shaft uh, or the hair shaft may be found. And this is called a follicular tag. This is what is used for DNA analysis and may be able to individualize the hair. The catagen phase is a transition stage of hair growth. It can last anywhere from 2 to 3 weeks. It continues to grow but decreases in the rate at which it is growing. The roots have an elongated appearance as the bulb is starting to shrink and be pushed from the follicle. And the telogen phase is the final growth phase. It lasts anywhere from 2 to 6 months. The root has a club-shaped appearance, hair as the hair is pushed out, causing it to naturally be shed. So hair is constantly going through this life cycle and process, and so especially during sexual assault cases where they comb the pubic hair and the head hair, they're trying to remove loose hairs that may be contributed to the suspect. This ends lecture 12-1. There's a quiz next class.